Good afternoon, everyone, and welcome to the 2022 Susan Swerling lectures. I am Nika Daniel and have the pleasure to serve as the organizer and host of this year's lectures focusing on cancer and metabolism. I am happy that the donor for these lecture series, Mrs. Phyllis Swerling-Kellen, and her family are also tuned in this afternoon. The Susan Swerling Visiting Scholars Fund was established by Phyllis Swerling-Kellen in 1981 in memory of her daughter Susan, who succumbed to ovarian cancer in 1975 at the age of 20. In Susan's memory and to promote Dana-Farber's life-saving work, gifts were donated to support an annual lecture where leading cancer researchers and scholars are invited to share their latest findings with peers. Phyllis is an honorary trustee of Dana-Farber, a dedicated member of Dana-Farber Society, and has volunteered at the front desk of Dana-Farber for over 40 years, serving with great energy and enthusiasm. Every year she attends this seminar with excitement and hope that the research presented will help save someone else's child. This afternoon, we have three stellar speakers who continue to do pioneering work at the interface of cancer and metabolism. We have time for discussion after each speaker, so please submit your questions via the Q&A tab and not via the chat tab, please. Our first speaker, Ralph de Benadinis, is Chief of the Division of Pediatric Genetics and Metabolism at the Children's Medical Center Research Institute and a professor at UT Southwestern Medical Center and an HHMI investigator. He holds the Joel Steinberg Chair in Pediatrics and is a member of National Academy of Medicine. We are also privileged to have Ralph as an external advisory member of our T32 training program in cancer chemical biology and metabolism here at the FCI. In addition to longstanding interest in metabolic reprogramming and metabolic liabilities in cancer, Ralph's lab has been at the forefront of developing methods to interrogate metabolism in intact tumors in living subjects. Ralph, thanks again for sharing your work with us.
Thanks so much, Nika. It's really great to be able to participate in this symposium and share some of our data with you. So hopefully the slides look okay. Here are my disclosures. And so my lab studies processes by which mutations cause disease through effects on metabolism, which you can see there on the left. And we study two kinds of disease that follow that paradigm: inborn errors of metabolism and cancer. So those diseases are very different, but in both cases, perturbed metabolism contributes to disease progression, and some aspects of altered metabolism are clinically useful either for diagnosis or in some cases for therapy. So I'm going to focus on cancer today. Cancer is a genetic disease, as you all know, and it's characterized by altered metabolism at the cytal level. And that's shown in part by the uptake of glucose analog FDG by this patient's lung cancer. Of course, this is an imaging modality that's widely used in the clinic. So why does that happen? Well, it happens because oncogenes, like the ones shown there in green, stimulate glucose uptake, lactate production, and other pathways. So metabolic reprogramming in tumors is in part related to the genetics of the tumor. And when I started my lab, there were two perceptions about cancer metabolism that I thought were interesting and that have inspired our work over the years. The first is that cancer metabolism is relatively homogeneous because all those oncogenes converged on a few important pathways like glycolysis, also known in this context as the Warburg effect, which is thought to promote cell growth. And the other was that activating glycolysis came at the expense of mitochondrial metabolism, and that mitochondrial metabolism might even be counterproductive for cell growth. And that seemed puzzling because usually mitochondrial pathways like the TCA cycle produce energy and building blocks and other factors that cells needed to grow. So on some level, we've been studying those two questions ever since then. And I'm going to address both of them during the talk today. The first simple question is, do all cancer cells use the same pathways? So we tried to address this a few years ago by culturing over 80 lung cancer cell lines under identical conditions in the tissue culture room. And then we used carbon-13 labeled glucose or glutamine to characterize metabolic activities. And because all the cells had been DNA or RNA sequenced, that allowed us to look for common factors in cell lines that had similar metabolic properties. So here's a graph, and each dot on the graph is one of the cell lines. And the plot shows the correlation between two labeling features in this metabolite, citrate, a TCA cycle intermediate and a precursor for lipid synthesis. So the two features on the two axes read out two different ways that cells can make citrate, shown by the blue pathway and the red pathway. And the position of the dot reflects how much each cell line uses one pathway or the other. And the important point here is that the cell lines are very heterogeneous, even with something as simple as making that central metabolite, citric acid, citrate. So in this case, the heterogeneity reflected the genetics, because EGFR mutations stimulate the blue pathway by phosphorylating and activating those enzymes, IDH1 and IDH2, responsible for that pathway. And the EGFR mutant cell lines are mostly found up here in the upper right quadrant. So that same kind of heterogeneity applies to most of the metabolic features we studied in that paper. And so the point is that even when cancer cells are happy, healthy, and growing in the dish, they use quite different pathways to do even simple things like making these central metabolites. But tumor metabolism can't be fully modeled in culture, because tumors have complex microenvironments composed of both malignant and non-malignant cells. And furthermore, all of these factors, in addition to genetic heterogeneity, can contribute to metabolic activity in cancer cells. And we don't have a good way to model all of those factors simultaneously in ex vivo models. So we realized several years ago that we had to have better ways to probe tumor metabolism in vivo, and preferably directly in patients. So for about 10 years now, we've been using intraoperative infusions with isotope-labeled nutrients like carbon-13 glucose to study tumor metabolism in patients. And I'm going to show you some data initially from non-small cell lung cancer. The way the study works is that when we identify patients that will have their tumors either biopsied or surgically resected, on the day of the operation, we infuse the patient intravenously with carbon-13 glucose, which circulates through the body, gets taken up by the tissues, and then the carbon-13 label gets allocated downstream into metabolites as a consequence of the metabolism happening in the body. So after a couple of hours of infusion, the surgeon removes the tumor, and then we can isolate the metabolites and measure carbon-13 labeling, both in the tumor and in the adjacent non-malignant tissue. And then over time, follow the outcomes so that we can correlate particular metabolic features with aggressive disease. And I'll show you some data about that in the next few slides. We've analyzed about 70 lung cancer patients that way so far. And this has all been a collaboration with Kem Kernstein, a great collaborator in thoracic surgery here. So we've, as I mentioned, analyzed several patients that were infused with uniformly carbon-13 labeled glucose. That is, all 6 carbons now contain the carbon-13 label, so that you can track the fates of all those carbons through the glycolytic pathway shown here, through the pyruvate dehydrogenase reaction into the mitochondria around the TCA cycle, and into other pathways as well. And here's what the aggregated data look like from the patients. This is a little complicated, but I'm just going to point out a few key features. The red dots are fragments from the tumor. The blue dots are a fragment from the adjacent lung. And these correlations are basically comparing labeling in one Krebs cycle intermediate to another Krebs cycle intermediate. Those should correlate strongly if the data are faithfully reporting the pathway. And I think they are, because of these strong correlations throughout the graph. The other important thing to realize is that there's quite a bit of heterogeneity, which you can maybe easily see here in the distribution of labeling in citrate or malate, et cetera. And there's heterogeneity both in the adjacent lung and in the tumors. But crucially, for what I'm going to talk about for the next few minutes, there's always a rightward shift or a shift towards higher values in the tumor, which means that carbon-13 from glucose in the circulation is a little bit better at getting into the TCA cycle intermediates in the tumor than in the adjacent lung. And that suggests that there's a significant amount of TCA cycle activity in the tumors. And I think even more importantly is the fact that when we correlated all of the metabolic features, several hundreds, both in terms of abundance of metabolites and labeling of metabolites, and asked which ones best correlate with outcomes, the ones that collaborated the best were labeling of TCA cycle intermediates. That's shown here, where the entire cohort is just split into tumors that had higher than average labeling and Krebs cycle intermediates versus ones that had lower than average labeling. And the Kaplan-Meier curve shows you that the overall survival is significantly worse in the patients whose tumors have more labeling. So if the tumor has more carbon-13 labeling in the TCA cycle intermediate, the patient does worse, and they usually almost always in this disease are dying because of distant metastases. So that's worth studying, of course, because metastatic disease is the main source of mortality in lung cancer and many other forms of cancer. So I'm going to spend some time talking about that too. But before I do that, I just want to mention there's a subtlety about TCA cycle labeling that is worth remembering. The TCA cycle shown here is usually coupled to oxidative phosphorylation because the cycle passes reducing equivalents to electron carriers like NADH. And then those carriers get recycled by the electron transport chain, okay, usually. It doesn't have to be that way because there are other mechanisms to recycle those carriers. And I felt like we needed to work this out because it affects how we might try to block the pathway. So Pani Paknis, a postdoc in the lab, grew xenografts in mice and performed carbon-13 glucose infusions with or without this drug, which blocks complex 1 of the electron transport chain. So if the electron transport chain is needed to get that labeling in the TCA cycle, then the drug should suppress the labeling. And that's what's shown here. Really nice suppression of carbon-13 labeling in mice that were treated with the drug. Okay, then Pani modified the cancer cells to express NDI1, a yeast protein that basically circumvents complex 1 inhibition. And that completely restored TCA cycle labeling. So what that experiment told us is that complex 1 in the cancer cells is enabling the labeling of the TCA cycle intermediates that we're observing in both in the mice and also in the patients. And that means that we could potentially use this compound to block that activity, which seems to be correlated with cancer progression. So from there, we need models of lung cancer metastasis to study this. And ideally, they'd be derived from the tumors in the patients where we observed that relationship. And that's turns out to be a big ask because it's been difficult to get patient-derived xenografts from non-small cell lung cancer to spontaneously metastasize in mice. Now, we were lucky to collaborate with Sean Morrison, who helped us use methods that he developed to assess metastasis in melanoma PDXs. So in this method, small tumor fragments are transplanted from the patients into the flank of immunodeficient mice, carried through one passage, and then the cancer cells are tagged with luciferase and then reimplanted. And that allows you to follow metastatic spread using bioluminescence or by assessing human HLA expression in the distant organs. Okay, so Brandon Faubert, a postdoc in my lab who ran this project for several years, implanted tumors from about 50 patients and got 11 of them to grow in the mice. 10 of the 11 came from tumors that had the desired high labeling pattern, and only 1 didn't, okay? And those tumors do metastasize. So here's 2 models showing the fraction of the mice bearing tumors in the flank that had viable human cells elsewhere in the body. And the fractions tell you that it's almost all of the tumors, all of the mice that have tumors in the flank are getting viable human HLA positive cells in the other organs, okay? With one exception, that exception is shown here in blue. That's the single PDX that came from a poorly labeled area of a tumor in a patient. Okay, that's a special case because it came from a patient whose tumor also had different labeling in different areas. So areas of high and low labeling from the same tumor formed PDXs in the mice, but only the tumors from the high labeling area in the mouse were able to spontaneously metastasize, as shown here. Okay. Now, sometimes these are more than just scattered viable cells. So here's a line that arose from a patient who developed a brain metastasis about 3 months after the infusion and surgery. Okay, and mice carrying those PDXs develop neurological conditions like incessant circling, like you see in this mouse here, constantly turning to the right. That can be associated with a brain mass, and in that mouse it was. You can see here by cytokeratin 7 staining that there is a mass that expresses epithelial markers in the brain of this mouse. Okay, so that set us up to test whether blocking oxidative phosphorylation suppresses tumor growth or tumor spread. So we implanted tumors in the flank as before, treated the mice with that complex 1 inhibitor that I mentioned, or a vehicle control, and then watched what happened. So at the dose that we used, the drug is well tolerated and it has a non-statistically significant effect on growth in the flank, but it does reduce the number of viable cells circulating in the blood. And more importantly, it reduces the metastatic burden in the 2 models that we've tested so far, shown on the left by detecting viable human cells in the brain, and on the right on a logarithmic curve here, by assessing bioluminescence in the lung. Okay, so note that we only have partial reduction of the ETC in the TCA cycle labeling using this dose of the drug. So it's possible that we could do somewhat better still. And really what we need to do in these models is to dissect the mitochondria genetically to try to understand which aspects of oxidative phosphorylation seem to be supporting this phenotype. But to summarize, so far, glucose supplies multiple pathways in human lung tumors. So the tumors are glycolytic, but they also variably use glucose as a respiratory substrate. And the ones that seem to be the best at doing that as a readout of labeling in the TCA cycle intermediates are the ones that progress the most quickly in the patient. Okay, now this approach is versatile. And so it's being used in many different types of cancer at UT Southwestern and now elsewhere as well, including this nice study from the Rabinowitz lab published last year. Okay, we don't have outcomes data in those other studies, but in all these tumors, there's evidence of oxidizing glucose and other respiratory substrates along the lines of what we found in lung cancer. Okay, and I didn't mention this, but I'll just say it here. Some of the labeling in the mitochondrial pathways arises indirectly through conversion of glucose to lactate elsewhere in the body, and then uptake of lactate into the tumor. And that too promotes metastasis, at least in some experimental systems. And we've published on that in a couple of different places, shown here and shown down here. Okay, now it would be reassuring, I think, to find tumors that do something different. So we decided to study clear cell renal cell carcinoma, the most common form of kidney cancer. Those tumors should look different, okay, because they mutate the tumor suppressor VHL, and as everyone knows, that causes constitutive stabilizing of HIF transcription factors, essentially causing cells in the tumor to behave as if they're hypoxic. The response to hypoxia, of course, is to activate glycolysis and suppress glucose oxidation. So we expected these tumors to be hardwired to have that kind of metabolism. And there's plenty of evidence from preclinical models, work from, I mean, among others, Bill Kalin, who of course won the Nobel Prize for studying the system, and other labs as well, that this kind of metabolic shift happens in preclinical models with HIFs turning on the expression of genes that activate that form of metabolism. And a key factor is turning on the kinases like PDK1 that shut off pyruvate dehydrogenase. That comes from work from Chi Dang and Greg Semenza and Nick Danko many years ago. So based on what I've shown you already, it should be really straightforward to assess that by infusing carbon-13 glucose in these patients. Divya Bezwada, a student in my lab working with Vitaly Margulis in urology, have been working on this for a few years now. And so after infusing carbon-13 glucose into over 50 patients with kidney cancer, the answer is yes. There's a clear suppression of labeling and Krebs cycle intermediates in clear cell renal cell carcinomas relative to the adjacent kidney. Assessed here is the relative amount of carbon-13 in citrates in the TCA cycle relative to the upstream metabolite pyruvate. Okay, so a nice suppression, about half on average. You can see they're still heterogeneous, but this is exactly the opposite of what we saw in non-small cell lung cancer, where the average gain in labeling was about twofold over the adjacent lung. Here, the average loss of labeling is about half of what you see in the adjacent kidney. Okay, so we wanted to know whether PDH suppression was sufficient to explain the labeling differences between the tumor and the kidney. And we approached that by infusing some of these patients with carbon-13 acetate rather than carbon-13 glucose. We did that because acetates can be converted to acetyl-CoA and enter the cycle without first going through pyruvate dehydrogenase. Therefore, should be a good test of that hypothesis. All right, now to describe the data, I need to make one other point about isotope labeling. I've presented all the labeling so far in this form. It's what's called isotopologues, or how many carbon-13 nuclei are present in a metabolite. So a metabolite like glutamate that has 5 carbons has 6 isotopologue forms, 6 labeled forms, and you could have anywhere from 0 to 5 carbon-13s, so you get 6 isotopologues. But actually, if you consider the position of those labels within the molecule, the sad news is that there's actually 32 possible labeled forms or isotopomers, okay? And M+2 alone comes in 10 different flavors. So if you were able to deconvolute those, you'd have a lot more clarity about metabolism, because as you can see here, the position of the labels change within the molecule as the TCA cycle turns over. You can see on the first turn, for example, in glutamate, the labeling goes into positions 4 and 5, but then it changes position as the cycle turns over. Okay, so we would like to be able to get all of that information together. Now, NMR reports many, although not all, of those isotopomers, and it's very insensitive compared to mass spec. So we want a sensitive method to detect as many of the isotopomers as possible. So Feng Sai, who's an analytical biochemist in my lab, developed a tandem mass spectrometry approach to painstakingly identify fragments of glutamates and also aspartate with the assistance of a computational method he developed that would allow him to detect all 32 labeled forms of glutamate and all 16 labeled forms of aspartate, as well as some metabolites related to other pathways. And that method works pretty well, as shown in the isotopomer patterns over here on the right, produced by cells cultured in the presence of carbon-13 glucose. You can see how the mass spec in orange performs relative to the gold standard NMR in blue for labeling patterns in both glutamate shown on the top and aspartate in the bottom. But the key thing is that Feng's method actually reports more information because he gets the complete set of isotopomers. And even more important, maybe in terms of sensitivity, requires just 1% of the tissue. All right, so now here's the data from patients with clear cell renal cell carcinoma that were infused with carbon-13 acetate. The total fraction of M+2 glutamate is shown here, and it's actually the same between the tumors and kidneys. But the positional information is really helpful, because in the tumors, most of the labeling is specifically in positions 4 and 5, indicating that it comes from the first turn of the cycle. So that indicates that with acetate as the tracer, the carbon is actually pretty good at getting into the TCA cycle, but it doesn't turn over. So in addition to whatever's happening at the level of PDH, turnover of the cycle seems to be low in the tumors. Okay, now you probably remember from before that for the cycle to turn over, the mitochondria respire. So we thought that poor turnover might indicate that the mitochondria aren't respiring very well. And the direct way to test that is to measure respiration in freshly isolated mitochondria. A tough experiment, but one that Divya was able to perform in collaboration with Nick Lesner. So she isolated mitochondria from the kidney and from the tumor from 12 patients, and then measured how well they respire or consume oxygen, basically in a seahorse assay. So it turns out it doesn't matter actually what you feed the mitochondria. They don't respire very well. And that means that there are more widespread problems in these mitochondria than just pyruvate dehydrogenase. Okay, and this is I think, the most reductionist way you could perform this experiment, because the mitochondria have been taken out of the body and out of the tissue and are now analyzed in isolation, where we're normalized for mitochondrial content between the kidney and clear cell, but you see this nice suppression of respiration. Okay, now maybe we should have guessed that. There's a nice paper a few years ago shown on the right from Ed Resnick at Sloan Kettering, describing a method that mines sequencing data to determine the mitochondrial DNA copy number. And what Ed reported is that clear cell tumors, shown at the top, have the least mitochondrial DNA when normalized to the adjacent kidney. Okay. And satisfyingly to us, the ones that have the most mitochondrial DNA are actually non-small cell lung cancers. Okay. So, so I think that makes sense with respect to the respiration data. But there was a statement in the paper that caught our attention that although mitochondrial DNA is depleted in these tumors, the amount of mitochondrial DNA was negatively correlated with outcomes. In other words, the patients who did the worst were the ones that had the most mitochondrial DNA. So in clear cell renal cell carcinoma, like lung cancer, mortality is largely determined by metastasis. So we went back to the infusions and compared labeling in primary clear cell renal cell carcinoma, still in the kidney, to ones that had metastasized outside of the kidney. So we've looked at 10 patients so far, and the data is shown there. And look how different the labeling is in the metastatic tumors relative to the primary tumors. They look much more like the lung tumors that went on to metastasize. Okay, so it's a different comparison, because here now, for the first time, we're comparing primary tumors to mets from the same type of cancer. But it suggests that in this disease too, the cancer type with, the cancer type with the least mitochondrial DNA, mitochondrial metabolism still seems to be associated with aggressive disease. So that makes it interesting. I think may be important to study whether mitochondrial activity is limiting for metastasis, and we've just now started to do that. NDI1, that yeast protein that I mentioned before, is useful because it allows you to do a gain of function experiment by just expressing the protein and driving ETC function and then examining the biological effect. Okay, so we've expressed NDI1 in VHL deficient clear cell renal cell carcinoma cells. Luciferase tagged them. Okay, expression of NDI1 is sufficient in these cells to double respiration and to triple labeling from carbon-13 glucose. And we're just now starting to assess the effect on metastatic spread. This is a simple experiment where the cells were injected directly into the tail vein, okay, and then we examine metastatic seeding in the lung. But I think what you can appreciate is that NDI1 expression has a pretty substantial effect on metastatic seeding and persistence of metastatic cells in the lung. At least in this model, the metastatic burden is at least doubled in this model. Okay, and so we still have work to do, of course, but I think that emphasizes the importance of electron transport chain function on cancer progression, even in cancer cells that are programmed to be glycolytic. So let me stop and sum up. Intraoperative carbon-13 infusions can be used to assess metabolism in tumors from patients with many kinds of cancer. Non-small cell lung cancers are metabolically heterogeneous in the body. High labeling in TCA cycle intermediates is related to cancer progression, and it seems to be required for metastasis in the PDX models that we've developed. Primary clear cell carcinomas display reduced TCA cycle labeling and oxidative phosphorylation, but from the data we have so far, the metastatic tumors seem to regain that ability to oxidize glucose, and we're examining whether that retention is important for metastasis. So there's a lot of outstanding questions. That maybe the most pressing one is, how does mitochondrial activity become reactivated in those metastatic clear cells? We have some hypotheses that we're testing there. What specific benefits of oxidative phosphorylation are important for metastasis? And then where along the complicated metastatic cascade are those benefits exerted? And so I hope that in the coming year or so, we'll have some answers to those questions. Let me stop and thank the people who contributed to the study. Most of these folks I mentioned along the way, I just make special mention of Ling Tsai, our informaticist, who helps us to think about the data in creative ways. We have a great collaborative team here in both lung cancer and kidney cancer. None of this would be possible without the patients and their families for participating in the studies. And I'd like to thank these sources of support. Thanks so much for your attention.
Thank you, Ralph, for a great talk as we are waiting for questions to come through Q&A. I was wondering what your thoughts are about how much of the heterogeneity, metabolic heterogeneity, for example, you're seeing in the lung cancers, how much of that are genetically encoded in terms of if you had gone back and looked at signatures from those same patients, whether there was a correlation?
Yeah, thank you. That's a great question, one that we've thought about a lot too. I would say the level of molecular analysis that we have now in the patient's tumors is sort of at the level of the main oncogenic drivers, KRAS, EGFR, handful of other genes, tumor suppressors, P53, STK11, et cetera. And it's been somewhat disappointing that we can't pin position on the spectrum of TCA cycle labeling on a particular oncogene. You know, the EGFR mutant tumors tend to be a little bit lower. The KRAS mutant tumors tend to be a little bit higher, but these are not black and whites. If you look at the RNA sequencing, you start to get a picture of a mixed signature in the tumors that have more oxidative features. So that may be contributing, although I don't think all of those tumors have amplifications of MYC for sure. So not a clear picture. I should say, you know, I pulled out the example of the EGFR mutants in a particular metabolic feature in the cancer cell line analysis at the beginning. But even in the cell lines, it's pretty difficult to get dichotomous metabolic features around a single genetic change. So this may mean that the ultimate phenotype is a consequence of combinatorial mutations. And I don't think we have the statistical power in the clinical study to assess that yet.
We have a few questions from the audience. What is the relationship between stromal and tumor metabolism? I think this is super broad, but your thoughts would be really appreciated.
Yeah, it is broad. It's absolutely vital. And in the field, okay, we've got to figure out a way to do more to deconvolve these issues. We are at the level now of basically doing high content, detailed, quantitative histopathology to try to correlate some of these features. You know, at the lower tech level, okay, basically just trying to assess, you know, gene expression signatures that would indicate the you know, the infiltration of certain cell types, say immune cell types. There's not a lot of signal that comes out with respect to central carbon metabolism. I think the NDI-1 experiment in the cancer cells at least tells us that electron transport chain activity in cancer cells themselves can dictate some of the TCA cycle labeling features. There are other metabolic features that correlate strongly with the presence of stromal components, including immune components, but we have a lot more work to do along there. This is very important. I think it's on everybody's mind.
Thanks. Bruce Spiegelman asked, are the PGC1 proteins involved in the modulation of OXPHOS in your systems?
Yeah, so Bruce, thank you for the question. I think, you know, I didn't show this, but one of the gene expression signature that correlates the best with labeling in the TCA cycle are basically the OXPHOS gene signatures. And so we have not looked to see whether PGC1 proteins are enhanced in those tumors, but something seems to be driving a gene expression program that includes the OXPHOS gene sets. And, you know, PGC1 alpha and MYC talk to each other. So my guess is that there's a MYC PGC1 alpha axis, but we haven't proven that yet. Now that we have these PDXs, I think one of the things we can do is to go in and try to silence that transcriptional network and see what happens to both the metabolic signatures in the tumor and also metastasis.
Nama Kanarek is asking, do you think the change in oxygen levels in the blood relative to the kidney itself is important for the role of OXPHOS in metastatic process?
It's possible, although I'm not sure. I mean, where the exposure to oxygen would be highest would be in the circulation. And I think data particularly from Sean Morrison's lab has sort of shown that too much exposure to oxidative metabolic pathways during the transit in the circulation is probably suppressive for metastasis, because there's a challenge for those cells to overcome the oxidative stress burden. So, it may be that there's some window during which in the circulation, where nutrient uptake is likely suppressed relative to what's happening in the primary tumor. Joan Brugge is the first person to show data supporting that hypothesis well over 10 years ago now, that the metabolic efficiency needs to be better in the circulation in order to maintain viability, at least until the cell regains a foothold at a distant site and begins to receive the signals that allow it to take up nutrients again. So it may be a question of metabolic efficiency more than just exposure to oxygen.
Do you see any differences in metabolic labeling in lung tumors that in the absence or presence of cachexia? And actually this question from audience remind me that perhaps this discussion could also move into the metabolism of the host and how you're seeing some of your labeling patterns and if there is information there.
Yeah, another great question, I think a pressing question in the field. We don't have enough non-small cell lung cancer patients that were frankly cachectic to be able to address that question. We're very interested. We have looked at issues like the effect of BMI on metabolic labeling in the tumor and haven't found much. The only thing that we found so far that correlates with BMI is the amount of labeling that we can achieve in circulating glucose. And I think that probably is just because some of the patients with high BMI have changes in glucose turnover, maybe are pre-diabetic, in some cases actually diabetic, so that it is it's harder to get the same amount of carbon-13 labeling in the circulation. And so that scores across the cohort. But so far, the metabolic activity within the tumor has not scored. And it may be that we're not looking at it in quite the right way.
Another question is if you think that the recovery of mitochondrial respiration occurs prior to metastasis, or does it help those cells adapt to their distant niches?
Yeah, I mean, I think we don't really know yet. I think the hypothesis that we are favoring is that there is a heterogeneous distribution of both the abundance of mitochondrial DNA and the kind of mitochondrial DNA in the primary tumor, and that what's happening during metastasis is selection for ones that maybe are a little bit more adept, either because they lack loss of function mutations that might be present in the primary tumor or some other factor that are just more adept at carrying out these oxidative reactions in the metastatic tumor. We don't know about homing yet. I have to say, particular predilection for any site that we don't know yet. I mean, I showed you the data that we have in the experimental models of metastasis in kidney cancer. Right now, it's simply tail vein injection and then looking at metastatic take in the lung. Even in the lung cancer PDXs that I mentioned earlier in the talk, I don't think we have much evidence for homing there. I showed you a couple of tumors that metastasize to the brain in the patient. And those same tumors can metastasize to the brain in the mouse, but they also metastasize to other organs. So I don't think I'm not sure we're set up to address that question in the right way.
There's another question that is actually close to my own heart too. It's asking whether you have tried ketone body labeling in patients.
Yeah, no, we talk about that a lot. I usually think of you with when we talk about that, either ketone utilization or fatty acid oxidation, because of your work in DLBCL many years ago, showing that there's a subset that's highly dependent on fatty acid oxidation. We haven't done it yet. I think ketone bodies are probably a more logical way to start in some ways than fatty acids because of ease of delivery. Yeah. And we should try. I certainly don't think we've accounted for all of the acetyl-CoA that goes through the TCA cycle by studying glucose, lactate, and acetate. So there are probably some nutrients that we're not accounting for. Fatty acids and ketone bodies would be the most likely.
Yeah. And do you see in any of your labeling studies that when there's multiple fuels available, the TCA pool of acetyl-CoA likes to be fueled by one versus another carbon substrate? Is my question clear? Or
it is. The question is just fuel substrate preferences, right? I mean,
yes.
I love the question because this goes back to Krebs's work.
Or substrate competition for a specific,
right.
Yeah.
I think we and others have some evidence through co-infusion studies that lactate may be special. It's really difficult to get a quantitative handle on exactly what's happening with these nutrients in an in vivo model, particularly in a patient. But under conditions where we think labeled glucose and labeled lactate are feeding the pyruvate pool equivalently, there's no question that the label that originates on lactate is much better at getting into the TCA cycle metabolites than label that started on glucose, even if the labeling in the pyruvate pool is equivalent. And so there's a mechanistic underpinning to that we're trying to understand now.
It's very interesting. And perhaps a last question on your finding about mitochondrial DNA abundance in your two cancer types, lung versus renal cell. Do you know what potentially can account for mitochondrial DNA depletion? Is it problems with mitochondrial DNA metabolism generally, or mitochondrial DNA distribution, or quality control, or mitophagy problems where mitochondrial DNA gets lost or not inherited properly from cells to cells as divisions go?
Yeah, it could be any of the above, right? Because since the mitochondrial DNA are handled separately, then they don't have a mitotic process like the like the nuclear DNA does. They get segregated somewhat stochastically during cell division, and that means that there can be drift in the mitochondrial DNA copy number. I mean, I think of mitochondrial DNA abundance basically following along the same processes that Bruce was asking about before, where you have a mitochondrial program that includes, among other things, replication of the mitochondrial DNA, creating more mitochondrial genomes. And so at least in lung cancer, where we think there's a connection to the abundance of MYC and the abundance of these mitochondria and the prevalence of these mitochondrial phenotypes, I mean, I think there probably is a drive on overall mitochondrial biogenesis that is driving up the mitochondrial DNA content.
Yeah, I think mitochondrial inheritance is also usually goes hand in hand with making just more mitochondrial genome. But again, those are all, I think, mechanistically poorly understood processes as to how they should be coordinated, but how they get coordinated. And perhaps our really last question here is more of a technical question from Irina Kareva, who's asking how technically difficult and expensive is to conduct these labeling experiments. Is it scalable?
Another question I've thought a lot about. I mean, you can't sort of whip this together over a weekend, I would say. It used to be that we would spend about $3,000 on carbon-13 glucose per patient, which is expensive, but it's not a total non-starter, especially if you're thinking about an experiment where you could get the information you needed with a small handful of patients, which is possible for some phenotypes. In terms of how technically challenging it is, I mean, once we have the tissue, analyzing the carbon-13 enrichment is no different than what we would do in a cell culture experiment. You extract the metabolites, run the mass spec. Interpreting the data takes a lot of thought. You can see how heterogeneous they are from patient to patient, even within regions of the same tumor. That takes a lot of thought. And then the biggest challenge is getting the team together, which includes the surgeon, the oncologist, the radiologist, the pathologist, the clinical pharmacologist, et cetera. And not to mention being compliant with everything the IRB wants you to do. And we're fortunate to have a good environment here to do these kinds of studies, but it's not trivial. I'd like to see other centers try it. We're always happy to help.
Yeah. Well, Ralph, this has been a wonderful discussion. Thank you so much for your thoughts, and thank the audience. I also thank the audience for their engagement. And just a housekeeping reminder, please type your questions in the Q&A, not the chat box. So we now move on to our, thank you, Ralph. We now move on to our next speaker, and my DFCI colleague, Pere Puigserver. Pere is a professor of cell biology at Harvard Medical School and professor of cancer biology at the Farber. His lab continues to do highly innovative work on fundamental metabolic and bioenergetic processes that influence cell survival and biologic functions. His longstanding interest in mitochondrial biology goes back to his original discovery of PGC1 alpha as a master regulator of oxidative metabolism during his postdoctoral training here at the Farber. And his research continues to delve deeper into both mechanisms and physiologic contributions of mitochondrial pathways in a variety of disease settings, including diabetes, cancer, and pathologies associated with mitochondrial DNA mutations. Pere, we look forward to your talk.
Please unmute yourself. Uh-oh.
Pere, I think you're muted.
He is muted. There we go.
Okay, excellent. Yeah. Well, thank you very much, Nika, for the introduction. And it's a real great pleasure to participate and to contribute to the seminars in oncology and the Susan Swerling lecture. And it's also, a nice pleasure to introduce or to present our work and to speak after Ralph, which is the work or the research I'm going to present. It's kind of good continuation along the same theme that deals with mitochondrial biology and cancer, which is one of the main processes and that our lab is interested. So, let me see. So as Ralph introduced well, to this audience, so mitochondrial organelles have an important role in cancer biology, both during tumor progression as well as in processes and resistance. And they follow and contribute the genetic evolution of the tumors, both in nuclear changes and mutations and genomic instability, as well as some mitochondrial mutation. As been alluded, the mitochondria has a DNA that follows also mutations which may be important for cancer progression. At the same time, mitochondria is responding to microenvironment interactions and nutrient as well as environmental conditions are going to impact the mitochondria and have impact on the tumor biology. So I'm going to go briefly to general activities that take place into these organelles. So as well know that the mitochondria is the main energetic hub of the cell, and as a consequence, it harbors reactions that are either at the membrane or are inside the matrix. And these enzymes carry out important metabolic as well as energetic functions. And this includes the oxidative phosphorylation, pyruvate metabolite, and TCA saccharides included in the previous talk, but also other metabolic pathways such as amino acids, the urea cycle, the fatty acids, ROS production, and then heme biosynthesis and redox reaction iron sulfur, and another pathway which is important for the redox control, which is 1-carbon metabolism. In terms of the mitochondrial biology, there is important cellular processes that have implications in cancer biology that includes the mitochondrial DNA replication that controls the mitochondrial DNA amounts. Also, all the dogmas of the cell biology as replication, transcription, and translation occurs inside these organelles that derives from bacteria. And also there is processes just the mitochondrial UPR that responds to protein folding responses, and also other proteins involved in the dynamics, fusion and fission proteins, other proteins and complexes involved in crystal biogenesis, and all the lipid transport and mitochondrial import that shape the mitochondrial protein and function. As far as cellular processes that these mitochondrial organelles contributes to the cell, and as a consequence to the organism organism of the, in this case are mitochondria harbors important cell death pathways which are important then for the survival. It also contributes metabolic to the proliferation as well as the growth and the cell differentiation process, redox. And an important aspect that has important consequences in tumor biology are the inflammatory senescence and immune processes that at the end I'm going to discuss. As Ralph has alluded, the mitochondria has been also thought to be targeted in cancer, and as a consequence, I just have listed in this case several targets of the mitochondria that involve proteins involved in glucose metabolism, as well as components at the phosphorylation, such as complex 1 and complex 3 and complex 4, and with mixed success, which probably deals with the mitochondrial heterogeneity that happen in tumors, as well as there are other compounds that perhaps not as specific have been brought to the cancer medicine and have with mixed success in tumors, and that might depend because we don't know exactly what are the targets. More recently, work here in this area coming from the Broad Institute that have generated this cancer dependency map. There are specific selectivities, particularly on mitochondrial ribosomal protein that are important for translation of these mitochondrial proteins, or in hotspot genes, and certain tumors seem to be highly or more dependent than others, and that might provide some vulnerabilities that can be explored together in some clinical studies. So I think the mitochondrial regulation, as we know from classical textbooks in cell biology, that occurs in normal cells are probably, at least at the first look, not that different in cancer cells. So the oncogenic driving mutations, I think, shape and contribute to the different functions of the mitochondria. But as far as we know as specific regulations, probably the mitochondrial biology that happens in these tumor cells follow the general principles of the mitochondrial biology that have been discovered in many different studies by different authors. And you want to look, what control the mitochondrial abundance in these cells. That might be dependent on the cell lineage specific, and that's again have been discussed a little bit in the previous talk. But one thing, or at least one component, perhaps related to the cell lineage, that is going to control this abundance is the differentiation state. As happened in normal cells, usually cells that that non-tumor and proliferate, they usually have less mitochondrial abundance, but as cells go through the differentiation process, they try to accumulate these organelles and try to be more dependent on oxidative phosphorylation. As an example, in tissues, the tissue that tend to be more susceptible to mitochondrial dysfunction they tend to be more differentiated cells, such as the neurons, the cardiomyocyte, or skeletal muscle cells that have these main pathologies in mitochondrial diseases. So perhaps the same kind of regulation of mitochondrial abundance occurs, at least as general principle, and there are exceptions, I'm sure, that can be discussed, and there are probably some different ways that mitochondrial abundance can be obtained. And the response to the mitochondrial mutations could be also similar. And then the mitochondrial adaptations, when mitochondrial is inhibited or depressed, that might be important for conditions of resistance. So in this lecture, I'm going to divide in 3 different chapters. So on 4 different chapters, sorry, and every different chapter contains important consequence for tumor biology. The first part I'm going to discuss is going to be the mitochondrial abundance and the cancer cell type that is mainly or can be mainly related to the survival. So the way I think is being set up by perhaps different studies, at least for normal cells, and at least in certain type of tumors. So usually tumors that tend to be more differentiated, they tend to have more mitochondria. More differentiated means that the cell lineage of the tumor perhaps express more cell markers which are originating from the tumor that the tissue is being formed. So just for example, in melanoma, that is one of the tumors that I'm going to discuss, like the MITF and some of the pigmentation pathways, is what defines, or what we might call, the differentiated state. And then the undifferentiated state, at least in general, I mean, it can be, I think, cases, but tend to be with lower mitochondrial abundance. And that's, we try to investigate this in the context of melanoma. And the way we start these experiments, we're looking at one particular coactivator that is responsible or causative of mitochondrial biogenesis, as Nick alluded. So this coactivator was discovered here at the Farber in the lab of Bruce Spiegelman, and many different work has put this coactivator, at least in healthy tissues or in normal tissues, in the control of mitochondrial biogenesis. And one important thing, at least in these non-tumor studies, is that the activation of PC1 alpha and mitochondrial biogenesis occur through many of these different transcription factors. And the activation of these transcription factors have 2 programs at the same time. One is mitochondrial biogenesis, but at the same time, there is a ROS detoxification pathway that mitigates or tries to decrease the oxidative state in terms of reactive oxygen species of these cells. So we probe certain or different tumors, and one of the tumors that we probe was melanoma. And these melanomas that we probe, it was approximately like 10% of the total human melanomas that express highly of this transcriptional coactivator. And this is also in melanoma cell lines or in short-term melanoma cultures. And as predicted based on the studies in normal tissues, this expression correlated with most of the oxidative phosphorylations and mitochondrial markers. And in these different cell lines, it was clear that inactivation of this gene in these high PC1 alpha melanomas and high mitochondria had compromised survival, and there was strong decreases in the cell number, which was not observed in tumors or cell lines that have not increased in PC1 alpha. Another vulnerability that these tumors had is that when we deleted a component of PGC1 alpha, which is the ER alpha. These high mitochondria, high PGC1 alpha tumors were very sensitive to the depletion of this ER alpha. And then in these two different cell lines, the tumor growth was decreased. At the same time, these tumors were having these defects on mitochondrial oxidative phosphorylation. One, I think, characteristic that this tumor had was an increase in oxidative stress. The ROS levels, as a consequence of the depletion of PGC1 alpha, and perhaps some decrease in this mitochondrial number, there was a burst in oxidative stress. And as a consequence, if we treat these tumors with a ROS inducer, such as the piperlongumine, then these tumors had basically no growth in xenograft models. So that means that at least in terms of the mitochondrial abundance in these cell lineage melanomas, the mitochondrial abundance and activity was required for the growth and survival, and at the same time was more sensitive to oxidative stress. And I'm going to go back, at some point into these different features. So the second chapter I'm going to move now, and also has been alluded a little bit, at least in abundance in the previous talk, is that mitochondrial mutations in cancer cells. So the mitochondria has this unique mitochondrial DNA, which is circular, that encodes basically certain proteins which are part of the oxidative phosphorylation system in different complex 1, the complex 3, 4, and 5, and at the same time encodes the tRNAs which are responsible for the mitochondrial translation. This, I think, is important, at least, or can be important, and has not been fully studied, for example, in conditions of the chromoform renal cell carcinoma, where they appear to have, in consequence, at least for some lineage of p53 deletions, are these ND5 deletions. And these tumors, as a consequence of this ND5 deletion, will completely have an inactive complex 1 activity. So in the context of the mitochondrial disease mutations, we have studied some cancer cell lines that have mutations in complex 1. And we have asked the question, what kind of vulnerabilities can have these cells? Or in other words, what kind of other genes can promote the survival of these cells? And we use CRISPR gain of function screens to identify the genes. And one characteristic of these cell lines that have these mitochondrial mutations is that they cannot grow in nutrient stress conditions or in galactose. So mitochondria is required in these conditions. If you have mutations in this, the complex 1, these cells are going to die. So one of the hits that we got in these tumor cells that have deletions or are deficient in complex 1 activity was the malic enzyme 1, which was expression of this enzyme was rescuing this complex 1 mutation. So the malic enzyme is a cytosolic enzyme which is responsible for generating NADPH, and is tightly connected to the TCA cycle, in a way that when the TCA cycle functions, then the citrate can be derived to the cytoplasm and used for lipogenic activity, and part of it can be used for the malic enzyme 1 activity, and in this case, it generates NADPH. So it functions, at least in this case, a kind of an antioxidant gene. And that might be important, at least in some cases, because Alec Kimmelman several years ago found that the KRAS human pancreatic ductal adenocarcinomas were somehow sensitive to this enzyme, and were requiring this enzyme for survival. So the reason why this enzyme can be important in this context is because of the NADPH productions. And what I'm getting is that sometimes, or at least in certain conditions, defects in the oxidative phosphorylation, in this case on complex 1, what is altering is all the redox state and then NADPH production, affecting the vulnerability of the survival in response to oxidative stress. And that brings to what is important, or what are the pathways that generate an ADPH, and that has been somehow very worked out by Rabinovich at Princeton, and at least in the cancer cell and in other cell types. And there is contribution from the one carbon metabolism, the malic enzyme, in this case, both cytosolic and mitochondrial, and then the pentose phosphate pathway. So in these conditions, we think that the malic enzyme can provide some survival benefits to cancer cells that may have mutations in complex 1. So we characterize these cells, these cancer cells, with mutations in complex 1. And if when they are submitted to conditions of glucose restrictions, these cells are very sensitive to cell death. But when the malic enzyme is overproduced in these cells, then the survival of these cancer cells can be somehow immortalizing the survival of these cells. And at the molecular level or mechanistic level, how then this complex 1 become sensitive to the malic enzyme, or how the malic enzyme 1 can rescue survival of these cells. So our model and our findings that I'm discussing here, or what we observed, was that in wild type cells or cancer cells that don't have complex 1 mutations, so when they are forced to glucose restriction, then in this case the pentose phosphate pathway is not producing an ADPH, because we are limiting the amount of glucose. So in this case, the mitochondria activates one survival pathway, which is the one carbon metabolism that derives from serine. And in this case, it generates sufficient NADPH that maintains the survival of these cells. So on the other hand, if you have a complex 1 mutation, and that brings me to perhaps these renal cancer cells, that they have these ND5 deletions, or any particular tumor cell that may undergo through complex 1 mutations. In this case, they cannot generate, in these conditions, an ADPH in the cytosolic. And this case, because of the mitochondrial complex 1 mutation, it cannot generate sufficient NADPH. So in this case, are basically relying on having sufficient malic enzyme 1 activity to generate sufficient NADPH that maintain the survival. That might become that in certain cancer cells with these complex 1 mutations, they may have vulnerabilities to this malic enzyme or other pentose phosphate pathways that could be used. And that might be also illustrated in condition in non-tumor cell, like the red blood cells, that they lack mitochondria. So they are very highly sensitive to oxidative stress and highly sensitive to mutations in enzymes that produces NADPH. So I'm moving to now the next chapter in this mitochondrial biology, and I'm going to discuss some recent work in our lab that define what's the role, or what contribution can have mitochondria to adaptations in tumor progressions, and at the same time, what is the role, or what kind of role can the mitochondria has in response to drug-targeted therapy. So in this case, I'm going to see, like in our melanoma model, what we have observed is that there can be a transition between these differentiated high MITF, high PGC1 alpha, that they move to undifferentiated melanomas that have less mitochondria. And in this case, they tend to depend less on oxidative phosphorylation. At least it's been a decline or less abundance. And in general, when we see this transition, we see increased oxidative stress. So when they lose these cell lineage markers, including PGC1 alpha, we see an increase in oxidative stress. So that's why we think it might be more sensitive to piperlongumine. And in general, and I don't know at what step these things, we see that these cells have more invasion and metastasis, at least in some models. And one important thing that we have found is that in this newer state, when they are more undifferentiated, where they try to produce more integrin and plasma membrane proteins, and at the same time that generates these new vulnerabilities. So in other words, the blockage of some mitochondrial pathways or mitochondrial regulators in these high melanoma cells, then they transition to another state which tend to be more resistant, but they're appearing some more new vulnerabilities that can be tractable perhaps. So in terms of the numerous progressions is that getting back to the studies that I presented in the first chapter, while we were observing that at least in the short term, the loss of PGC1 alpha and this mitochondrial program in melanomas was kind of transient. So at the beginning, it was a strong suppression of cell growth and survival. But after 2 or 3 weeks, then we were trying to appear that the rate of these cells were trying to be at the same time similar. So to some extent, what we observe is that it was the classical shift that is being shown in yeast biology, for example, is that when you inhibit the mitochondria, then the glycolytic pathway turns on. And at least in our system, one of the pathway that was turning on and maintaining the survival of these melanoma cells was HIF1 alpha, this hypoxia-regulated factor. And consistent with this activation, the glycolytic pathways were activated, and these cells were able to survive both in suspension as well in the dish and in the xenograft. So one characteristic thing that we have observed, and not only in tumor cells, but in other cells, like in these mitochondrial disease mutant cells and in other cells, as soon as when we perturb the mitochondria, when we make cells that they have deficient of mitochondria, these cells respond by increasing metastatic programs, such as programs in the integrin pathway, the focal adhesion pathway, the adherent junction, TGF-beta pathway. It's a kind of resistant state that maintains survival even in suspension. And interestingly, and we don't know at what stage these cells, so these cells have more migration and invasion, where they're being tail vein injected into the mice. So these high differentiated cells, they tend to be less, very less metastatic. But when we decrease PGC1 alpha and consistent with an increase in this integrin pathway, these cells become quite invasive. And we see this in tail vein model and then in the orthotopic xenograft models, in that we see massive invasion in this case. So we try to model now these resistance states, or this low PGC1 alpha, if it has any implications in human melanoma patients. And we collaborate with Whitlow Hans and trying to look the expression of PGC1 alpha in melanoma patients that have undergone through the BRAF and treatment. And what we compare is that what happened to the PGC1 alpha expression in clinical specimens that before treatment and after treatment, when these tumors are resistant to the BRAF inhibitor therapy. And we observe in that, so when we compare that most of the melanomas that pre-treatment were having high PGC1 alpha, at the end of the post-treatment that or on treatment, then these melanoma, they try to lose over the BRAF therapy the expression of PGC1 alpha, and that correlated with survival of these patients. So when they transit from the pre-treatment from high PGC1 alpha to low PGC1 alpha, it was an increase in death of these patients, and tumors that were not going through this transition, that they maintain the expression of PGC1 alpha, they try to, or they survive longer. So we try to model this in culture, and in different melanoma cell lines to try to define perhaps some vulnerabilities of these cells. And we went to different melanoma cell lines in which they were become resistant or adaptive to the BRAF treatment. And consistent with previous work, the early response to this drug-targeted therapy, the BRAF and even MEK inhibitor, in both cases, we see an increase on PGC1 alpha and mitochondria. There is a correlation on that. But then over time, most of the cell types, the cell-resistant cells, were losing PGC1 alpha. And at a certain times, then they were an increase then on cell number, but PGC1 alpha remained suppressed. And we think that this suppression tend to be epigenetically. So there is H3K27 that basically responsible for this suppression. But our interest was to try to identify vulnerability. So what happened to these cells that now have become resistant to this drug-targeted therapy? And we performed a metabolic screening, looking at metabolic drugs that were affecting the cell growth. And the top drug that was killing preferentially these resistant cells were statins. These are cholesterol lowering drugs. And we were very interested because these are clinical approved drugs and tried to investigate why these melanoma cells that become resistant to the BRAF and MEK inhibitor treatment, they are sensitive to statins. And the sensitivity was dramatically in, almost more than 8 or 9-fold in these cells. That could be mimicked by the deficient of PGC1 alpha, that these cells, if we remove just PGC1 alpha, can provoke the sensitivity to a statin. And in the tumor xenograft, we saw similar effects. So I'm going to have a brief summary of this result because there is a lot of mechanistic details here, but it turns out that the sensitivity of these melanoma cells, which are resistant to the BRAF, is not depending on the cholesterol branch of the statin effects. So there is the statin effects on cell growth, on cell survival in this case, is working through a RAP proteins, which are in the geranyl pyrophosphate or prenylation pathways that have to deal with trafficking of vesicles to the plasma membrane. In fact, when we compare the BRAF resistant and parental cells, the main cluster of proteins that were upregulated were plasma membranes. So it seems that when we suppress PGC1 alpha that induces this de-differentiation of these melanoma cells, the cell respond by increasing and upregulated the same pathways that we observe in the depletion of PGC1 alpha. So there's a very good correlation. And we think that these plasma membrane proteins and integrins and the cell addition is what maintained its cells, the cell survival. But why these cells were sensitive to these RAPs? It turns out, and that's probably my favorite slide of this lecture, is that that reflects a proteomic signature of a parental cell line, melanoma, that is resistant to a statin. If you look at the protein, this is like a fingerprint of the proteomic of this cancer cell, that there is almost one single cell that changed dramatically, which is the HMG CoA reductase that, as predicted, is the target of the statins in the cholesterol pathway. But if you look at a sensitive cell, look at the proteomic signature, a lot of proteins move to the right and left. So it's like a bullet that basically destroys this cancer cell, kind of displacing the proteins up and down. If we look at the proteomic then signature, that what are these proteins? These are proteins involved in the chromosome segregation, in DNA damage, in cell death, which are basically activated by statins. This can be just rescued by one single metabolite, the GGPBP, and then we can rescue this sensitivity or resistance by just expressing two different RAPs, the RAP6B and RAP27A, that we found that just ectopic expression of these two proteins rescue the the survival of these proteins. And this correlates with integrin and FAC signaling. So when we have resistant cells, our statin strongly suppress FAC signaling, and that can be mimicked by PGC1 alpha depletion, so exactly the same. So deleting the single gene, and then by overexpressing the RAP6B and RAP27A, we rescue this expression. So similar effects in the resistance. And at the more cell survival, we see that these cells are not surviving in cell suspension or the dish, so there is no growth or survival. And then just expressing these two proteins, we rescue the survival of these two proteins in this case. So I think what we have identified, at least in this case, is that when we switch the differentiation program, that at least in this particular case coincides with changes in mitochondrial abundance, there is an increase in oxidative stress. Cells become more vulnerable to ROS inducers. There is an increase in plasma remodeling and invasive states, which is the integrin and FAC signaling. But at the same time, appears that these resistant cells have new metabolic vulnerabilities, in this case to the statins, which is independent of the cholesterol pathway and more depending on the prenylation pathway that provides some vulnerability to these cells. And in this case, there is another vulnerability that I have not time to talk, that in this differentiated cell, it also become very sensitive to the ferroptotic pathway. So I'm just going to slide 2 slides on the mitochondrial immunity. I'm sure the next talk is going to provide more insights in different aspects of that. But it's in that the same pathway that we have working in other tissues and in cells, such as the PGC1 alpha and mitochondrial abundance, seems to also play a role in immune cells. In this case, we have most of the chapters have been talked about the tumor cell per se, but there is an important component of the mitochondrial biology in these immune cells. And in this case, in 2 different aspects, this coactivator has been to be important to maintain the metabolic activity in the tumor microenvironment in these T cells and to maintain the tumor survival. But also there is effort from different groups in trying to manipulate this coactivator and mitochondrial biogenesis and activity in the T cell therapy. And from our side, we have been lately interested in trying to investigate and move into what's the role of mitochondrial immunogenicity. And I'm not going to have a lot of data here, but just one of the experiments that we have done, we have used these mouse melanoma cell lines that we have inhibited the mitochondrial complex one, and using the inhibitor that Ralph has alluded, that has a strong effects in his tumors affecting the cell growth. In our mouse models, the tumor, when we look at the activity in the newt, has some effect, but is not statistically significant. The high statistically significant effect that we see is that when we have an immunocompetent competent mice, and we think that provides at least perhaps some vulnerability, and we have some additional experiment I'm going to present that affects some immune pathways that may also contribute to the tumor growth. And that goes along that these the mitochondria, in addition to this complex one in the immune cells, the mitochondria itself, because of the bacterial origin, it has many impact on the mitochondrial immunogenicity coming from the mitochondrial DNA, the formyl peptides on translation, and also harbors most of the inflammasome that can be important then in interferon signaling. So that's a conclusion slide. So I have, I think, provided some perhaps principles, in some case can be maybe tangential to some of the programs of mitochondrial biology and cancer, but I think I have touched, different aspects of the mitochondria in abundance, mutations and vulnerabilities, adaptation, resistance, and like immunogenicity that I think can provide some newer guides and newer foundations to study more in depth the role of mitochondrial biology in tumor progression, not only in the tumor, but also in the immune system, and also in the communication between the mitochondria in the tumor and the interactions with the immune cells. With this, I'm going to close the the lecture, and then to acknowledging people in the lab that have been involved in this cancer mitochondrial biology, particularly Chilu that started most of this work, but also Jason that has done most of the work on statins and in the immune. And then our collaborators in melanoma, longest time collaborator, Hans Winlot. Then with Steve, we have done most of the proteomics and some of the immune project that have had a chance to basically to introduce a lot. We collaborate with Kai in our institute, and some of the initial work on the CRISPR screens and identification of melanoma that was done from this folks at the Broad Institute. And this is some of the funding that we have here at the Dana-Farber from the different NIH institutes and the internal programs at the Farber and the Department of Defense and some other foundations. And I thank again to Nika and the organizers of the Swirling Talk for giving the opportunity to present this work. Thank you.
Thank you, Pere, for a great talk. We have a few questions. I'm going to start with one that asks or discusses that PGC1 alpha function is contradicted in different studies. It stimulates cancer metastasis in breast cancer, but suppresses metastasis in prostate cancer. Do you think PGC1 alpha is an oncogene or a tumor suppressor gene?
So that's a good question. So I think from our work, mainly it tends to be the survival gene, at least, and it tends to follow more the differentiated state. The more I think you have the PGC1 alpha, the cells are more survival, and that fits with all the biology and work that has been known in non-tumor cells, that PGC1 is protective and somehow anti-inflammatory or anti-ROS, or protects against oxidative stress. So now in tumors, I think some of these studies I think are problematic in a way that they are very, I wouldn't say is not very common that PGC1 alpha is highly abundant or highly expressed in a lot of tumors. So I think melanomas is one of the examples, and it might be certain tumors that it may have, high expressions. So I wouldn't say that this is general in all the tumors that they have this high expression. It could be that sometimes that MYC that tends to favor the mitochondrial biogenesis can work with other factors. So that's what I would say. To the question of whether then PGC1 alpha could be then in some cases controversial, I think the way I see is that first of all, the low expression can be problematic then, to analyze or technically to probe the PGC1 alpha. But in some other cases, it's a very highly inducible protein that it changes quickly, and the transiently and adaptability of the cancer cell is it can reprogram quite easily to different states. So I think that in our case, you decrease, you get transient decrease, but then it goes back. It's similar that happened with the drug target therapy. So it goes up, but suddenly the cell can suppress and then move on to a different pathway.
Another question relates to EMT. The person asked, EMT is highly related to metastasis and may induce mitochondrial dysfunction. On the other hand, your results seem to suggest that mitochondrial dysfunction can cause metastasis. My question is about the rational causality between mitochondrial dysfunction and metastasis.
Yeah, so I would be cautious on that. So why we present, at least with the PGC1 alpha, it seems that the association with mitochondria and metastasis is not as straightforward. So decreases PGC1 alpha, you see decreased mitochondria, but at the same time, at least there is another transcriptional pathway that links more with TCF transcription factors and the YAP pathway that have published that likely independent of the mitochondrial effects can drive the PGC1 alpha effects on metastasis and invasion. So I think I'll be a little bit cautious on that. On the other hand, what we have seen from mitochondrial mutant cells is that these cells survive better in suspension, at least in normal conditions. And there is, I think, a signaling program from the mitochondria to the secretion pathway that tend to be important for survival. To what extent this is important in the tumor types or in what kind of a scenario that is important, we don't know. I think, we may study.
We have a question from Ralph asking if the CRISPR screen provided any insight into where the malate for malic enzyme comes from. Did components of the malate aspartate shuttle or the citrate malate shuttle also score in the screen?
No, I mean, at least they were not sufficient by gain of expression to score in this screen. I mean, the other components were related to the ROS pathway, thyroidauxins and peroxyredoxins. So in the other one, we might, or at least in some cases, mitochondrial IDH, but that was not from the screen. That was after we probe some of the contribution of the IDH and TCA on an ADPH production, but that was not from the screen.
Bush Wiggelman asked if can the tumor vulnerability be exploited by inhibiting OXPHOS through inhibition of ERRs?
Yes. So actually, they've been certain companies interested in targeting ER alpha antagonist to the tumors. So one of the main issues, and I'm sure from the previous talk, can be at least indicated on that, is that, and at the end with the immune regulation, I tried to emphasize this, and that was one of the problems with these inhibitors, is that if one inhibits the mitochondria in the tumor, also inhibits the mitochondria in the immune cells, and T cells tend to be particularly vulnerable to that. So that's been some, I think, caution, at least in some of these companies, to move forward these ER alpha antagonists that have been used. From our studies, it was clear that there was a clear dependency depending on the melanoma type on ER alpha. But at the same time, there is work in immunocell that ER alpha is important for T cell function. So one may take into consideration that.
We have a question about how you characterize differentiated and de-differentiated mitochondria in cancer cell. Is this based on aggressive nature of cancer cells, differentiate mitochondria cancer cells, or de-differentiated mitochondria cancer cells?
No, I think, I'm not that knowledge on other tumor types, and we have been mainly most of our work has been in melanomas. And in melanoma, it's clear that the differentiated ones are clearly MITF positive, which MITF is the oncogene, and that drives the differentiation. And then that's linked also to a lot of the pigmentation pathway. And at the same time, you have the PGC1 alpha that, at least in a subset of the MITF, you get also these mitochondria ones. So I think in this case, in the melanoma, I think it's quite clear how to separate that. In other tumors, I would say perhaps in breast, one can do more of the ER transcription factors or some other cell lineage specific factors that can have more identities of the original tumor type or tissue type.
Another question is, the person is wondering why cancer cells with low amount of mitochondria show tendency to metastasize. These cells seem to grow slowly in primary sites, as exemplified by PGC1 knockdown in xenograft studies. How do you explain low growth advantage makes cancer cell easily metastasize? Thank you.
Yeah, that's a great question. So I think maybe I didn't make myself clear on this slide, and I think because it was temporally different during the presentation, is that I would say that the cell growth is temporal, like in the acute ways or in several weeks when you remove PGC1 alpha, the cells kind of collapse, but after several weeks, it's cell rescue, and when they rescue, they have the same amount of cell growth, at least, without rescuing the mitochondria, without rescuing PGC1 alpha.
And then as a probably last question, we have more of a broad, perhaps conceptual question as well. How different are metabolic and mitochondrial abnormalities between what happens in cancer and what happens in aging?
Yeah, I think that's a great question. I think that might depend maybe on or my intuition, and again, so I would say just intuition, and I don't know how much rational. Sure, there is not much of scientific data. What I would say is that there are a couple of pathways that may be a kind of similar, although maybe cancer is maybe more strong or maybe more aggressive in this case. So in the aging, we get all of these senescent inflammatory pathways. So in this case, one might consider that tumors with high degree of inflammation or hot tumors might be similar to that. At the same time, it could be that similar, tumors that have, high or elevated rates of mutations in the mitochondria, they might generate, at least some vulnerabilities, but also may have some of the aging phenotypes. So that's from the tumor perspective. I mean, another angle is like other groups are focusing even here and other places, that is the opposite. And you have mentioned at some point is how then the aging is affecting the tumor per se. But that's completely speculation that we don't have clear picture.
Yes. Perry, this was great discussion and a wonderful presentation. Thank you again.
Thank you, Nika.
And we move on to our last